So now I will invite Sharjeel Khalid from International Islamic University, and he is currently employed at NRC. Welcome, Sharjeel.
Thank you, ma'am.
So now I will invite Sharjeel Khalid from International Islamic University, and he is currently employed at NRC. Welcome, Sharjeel.
Thank you, ma'am. Today, my presentation topic is on safety analysis of microorganisms based on whole genome sequences. So, Bismillahirrahmanirrahim. I'll start our presentation. So, EFSA is the European Food Safety Authority, and they provide some guidelines about the safety analysis of microorganisms based on WGS. So, EFSA have basic guidelines about the safety analysis of microorganisms to be used as a food supplement based on WGS, and we will be discussing next. So, microbial food supplement. Pre-market authorization process must be used for the use of any microbial species in food feed supplement. We have to see the possible risk assessment to allow the microorganisms to use the food feed items. The use of whole genome sequence-based data has been established as a requirement for the risk assessment. WGS research data obtained from WGS whole genome sequence analysis can provide information on the microbial identification. WGS also provide information on the characterization of microbial potential functional traits, and these includes virulence factors, resistance to antimicrobials of clinical relevance for host, production of non-toxic metabolites, or their pathogenicity or toxicity. The microorganism tested under analysis should be the one subject to the application for authorization. The culture sample used for DNA extraction, WGS sequencing, data analysis, and the results reported should be clearly described. Pure culture, each microorganism should be cultivated before DNA extraction as a pure culture, and standard DNA extraction protocol should be applied. Total DNA that is including chromosomal and extra chromosomal elements should be extracted and subject to analysis. Whole genome sequencing step. Second step is library construction. DNA fragmentation method and selection of fragments should be reported. Any selection of fragments by size should ensure that small plasmids are not lost. The applicant should describe the sequencing strategy, instrumentation used. For short read sequencing technologies, it is recommended to trim the sequencing read. The average read depth achieved should be at least 30-fold with the recommended target of 100-fold. Sequencing strategy continued, that is, contamination in the sequencing leads should be investigated. Assigned leads to an unexpected organism should be less than 5%. If this is not the case, then the applicant should provide an explanation. The sequencing read can be de novo assembled and annotated, mapped to a reference genome database. Complete genome sequence should be pursued, but draft genome sequence may be accepted. If a de novo assembly based approach is taken, then the following data are required. De novo assembly including assembler software, version, and parameters. If post assembly processing is carried out approach, software version and parameters should also be reported. If a genome annotation is carried out to provide any of required information, the software name, version, and parameters should be reported. The database version where available and all data accession should be indicated. Use of WGS characterization of microorganisms. First is identification of microorganisms. Confirmation of the identity of microorganism under assessment should be provided. Identification is continued. For bacteria, the identity of organisms under assessment should preferably be established by digital DNA hybridization, every nucleotide identity, or phylogenomic methods. The data from the microorganism under assessment should be compared with the reference genome of the type strain of expected species. In case the genome of type strain is not available, another well-identified strain can be used as a reference. For yeast and filamentous fungi, confirm of identity should be done by phylogenomic analysis. Identification of microorganism continued. A phylogenetic tree is recommended in taxa where a high level of identity between related species exists. If reference-based read mapping approach is used for identification, sequencing reads should be mapped against a suitable reference genome. The choice of the reference genome needs to be well considered, justified, and reported. Genetic characterization of the genetic modifications can be comparing the WGS data of the genetically modified microorganism with that of non-genetically modified reference genome, parental or recipient strain. De novo assembly or strategy additions and small modifications, for example, regulatory elements, a reference-based read mapping approach can be used. For other genetic modifications, a de novo assembly or confirmation of the test strategy may be needed. Identification of genes of potential concern. The WGS data can be interrogated for the presence of genes of potential concern, which may include those coding for a contributing to resistance to antimicrobials relevant to the use in humans and animals. Others coding for virulence, pathogenicity, or toxigenicity. Identifications of genes of potential concern and include a de novo assembled sequence can be analyzed with the search comparison based approach against maintained databases, and the identified hits should be provided in the table. For each reported result, the subject sequence, that is the sequence in the database, name and accession number, function of the encoded protein, sequence identity, and the percentage length of the subject sequence which is covered should be provided. If a reference-based read mapping approach is used, the sequencing read should be compared to the maintained reference databases. The following statistics should be reported along with the subject, sequence, name, and accession and function of the encoded protein, sequence identity, the average depth of mapping, and the percentage length of the subject sequence which is covered by read. The strategy, software, and all relevant parameters, including algorithm, is specified within the software used to identify genes of interest should be reported. Antimicrobial resistance. When the search for antimicrobial resistant gene is required, it is recommended to conduct it against at least two maintained databases. The search should be done applying minimum available threshold in the database for the length of coverage. In addition, in case of microorganisms for which no or few AML genes are present in databases, searches with hidden Markov model tools are recommended. In general, query sequence hit with at least 80% identity at the protein level or nucleotide level as reported in the database, and 70% length of the subject sequence should be reported. In the case two or more fragments covering less than 70% length of the subject sequence with at least 80% identity to the same AMR gene are detected, these should be reported and it should be checked whether the full gene is present or not. Toxicity and pathogenicity. Depending on the taxon, the assessment may require the search of genes coding for non-virulent factors, for example, toxins, invasion, and adhesion factors, or to identify the presence absence of known metabolic pathways involved in toxigenicity. The search should be done applying the minimum available threshold in the database for the length of coverage. In general, query sequence hits with at least 80% identity at protein level or nucleotide level as provided in the database, and 70% length of the subject sequence should be reported. In the case two or more fragments covering less than 70% length of the subject sequence with at least 80% identity to the same gene are detected. These should be reported and should be checked whether the full gene is present or not. Thank you all. Thanks for your cooperation.
Thank you, Sharjeel. That was an informative presentation from your side.